So my name is Parkkinen. I'm a director of the Oxford Brain Bank and associate professor in neuropathology. So that means that I spend my days looking down the microscope, diagnosing who has changes that have to do with Parkinson's disease or Alzheimer's disease or some other type of dementia. But my group is also very interested in developing biomarkers, molecular biomarkers. And I'm thrilled today to be able to tell you about one of our success stories, which very much has to do with the first developing these tests by examining the human brain. But I will start by just introducing to some of the basic concepts of Parkinson's disease research in a very layman terms, and then I'm going to try to explain to you why do we need biomarkers. It's probably as important as really finding that cure is to find the biomarker that would be sensitive and specific. Then I will move on to the actual seed amplification assay that we call this protein test, which is the novel diagnostic test that you can do by examining a person's CSF, cerebrospinal fluid. And finally, I'll finish by explaining why the brain donations are still so important. So what causes the symptoms of Parkinson's disease? So the motor symptoms, the tremor, slowness of movement, and the rigidity are thought to be caused by a loss of dopamine-producing neurons in a very specific spot of your brain called substantia nigra in the midbrain. This is in the back of your brainstem. But a lot of neurons are also lost in other hotspots that are shown over here in red. These hotspots really do exist throughout your brainstem and also in the basal forebrain. So my message really is that sometimes I think we, because of levodopa treatment and it working so well, we are sometimes quite dopamine-centric. But I think we also need to focus our research into loss of these other neurons in order to really be able to treat all the non-motor symptoms. In addition to this loss of neurons in these several different structures, in these nuclei, we also have a alpha-synuclein protein, which is a normal protein, quite a small protein in a brain, starting to aggregate and form inclusions that I can see down the microscope inside the neurons, and these are called Lewy bodies. And these Lewy bodies exist and spread throughout the brain in certain vulnerable regions. And we don't quite understand yet why are first of all, the neurons lost in these specific nuclei, and why these Lewy bodies are spreading throughout the brain into certain vulnerable regions. But it's quite clear that Parkinson's disease is much more than just the motor symptoms. The motor symptoms, you could kind of see that that's just the tip of the iceberg. That is what we see. Whereas there's a lot of invisible symptoms. Starting from anxiety, there's autonomic symptoms such as constipation, we have loss of smell, and most importantly, what we have heard today already, we have this REM sleep behavior disorder, which is a loss of REM sleep muscle atonia, which means that you act out your dreams, and you can hurt yourself or your partner by doing so. But Parkinson's disease, it really, even though we talk about the disease, it is a syndrome, and it really is a personal journey for everyone. And it's a combination of different symptoms. Now, Parkinson's disease includes into a larger group of diseases, and these are called synucleinopathy. So this is just to do with the pathology that we can see. And these diseases can be roughly divided into 3 different groups. So these Lewy body disorders, which have those Lewy bodies that I just showed you. Then the multiple system atrophy, which is very similar to Parkinson's disease. It can be very difficult to Michelle to tell that apart. And then the prodromal disorders, which are something that you see some symptoms, but you haven't yet converted, and you can convert either to the Lewy body disorders or the multiple system atrophy. Now, the Lewy body disorders include Parkinson's disease and Lewy body dementias, if the Parkinson's disease is also characterized by additional dementia. And these dementias, Lewy body dementias, can be divided into two different categories. We have Parkinson's disease with dementia, if the dementia occurs after 1 year or start of the motor symptoms, so later on. And then we have dementia with Lewy bodies, if the dementia starts together with the motor symptoms. So you have a syndrome that has dementia and motor symptoms and visual hallucinations starting from the very beginning. Now, it's important to understand that when I look down the microscope, it's completely impossible for me to tell these apart. Parkinson's disease, dementia with Lewy bodies, and Parkinson's disease with dementia. But when I see a case with multiple system atrophy, I can see that straight away. And the reason for this is that I can see the aggregates not in the neurons, but in the glial cells, which are the supportive cells that support the neurons. And the pathological picture is quite different. But clinically, as I said, it's really difficult to tell this apart. But it's important for us to be able to tell these apart because most likely these will have to be treated in a different way. And then we have the prodromal symptoms, the pure autonomic failure, which is as it says, a failure of your autonomic system. And this is characterized by orthostatic hypertension and other autonomic dysfunctions, mainly to do with your urinary and GI tract. And the pathology in these is only seen in the periphery, in the peripheral ganglia. So you don't see anything in the brain yet. But it's thought that these patients can then convert to either multiple system atrophy or Lewy body disorder. And then we have the isolated REM sleep behavior disorder with that impaired REM sleep muscle atonia, which can also is thought to convert to multiple system atrophy and Lewy body disorders. At the moment, we don't know much about the pathology of this REM sleep behavior disorder because there's not that many autopsy studies. Now, another important thing to understand in order to understand where the therapies are going is that at the moment there's a lot of experimental and human evidence to think that this alpha-synuclein protein that aggregates, which a lot of the disease therapies, the modified disease therapies are trying to target, that this wouldn't happen, is that this alpha-synuclein, it can aggregate in different ways. It doesn't always aggregate only in one way and you have only one type of fibrils. And this is the strain theory, so that one amino acid sequence might fold into these distinct amyloid fibrils. And at the moment it's thought that these different diseases that I've just showed you, they cause the difference in age, and the strains cause the difference in age and onset, the clinical symptoms, the alpha-synuclein inclusions, whether they're found in those neurons, in Lewy body disorders, or in glia, in multiple system atrophy, and then also the neuropathological spread of these inclusions. How far do they spread? How much do we have? But it's to do with these strains, and that's at the moment a really hot topic to research. Now, another important to understand is as well that alpha-synuclein doesn't only exist in the brain, it also exists in the gut and other peripheral systems and biofluids, such as CSF and blood. And is considered at the moment that this dual hit hypothesis, the alpha-synuclein pathology could actually spread to the brain either through your olfactory bulb or through your gut, through the vagus nerve. But again, there's a lot of experimental data to show this, so animal models, but the human data is still something that needs to be established. But alpha-synuclein pathology is seen in all of these different peripheral biosamples. It's prevalent in skin, olfactory mucosa, in your heart, gastrointestinal tract, adrenal glands, salivary glands, and even in your eye, and it's very prevalent in blood, and also it can be detected in CSF, which is very much the basis for our biomarker, why we want to do this, we want to examine this alpha-synuclein in this medium. And it could have also therapeutic implications, because if we could detect it already before the alpha-synuclein would spread into the brain, we could then also stop it before this happens. Now, why did I say that the biomarkers are as important as finding a therapy? Is because of this fact, which is true to any neurodegenerative disease, is that the neurodegeneration starts already 10 to 20 years before the first motor symptoms appear. And this premotor phase is characterized by RBD, hyposmia, loss of smell, and constipation. But the problem is that if you only use the time of clinical diagnosis and start to treat at this stage, the disease process most often is too advanced for therapies to really have a full impact. And there is a significant error of 20 to 40% even. Very skilled movement disorder specialists, such as Michelle, do get it wrong, because some of the cases do end up being, for example, MSA. And at the moment, the post-mortem verification, what I do, looking down the microscope to find those Lewy bodies and looking at where the neural loss happens, it really remains still, unfortunately, the gold standard for the definite diagnosis. So what do we want with the suitable biomarkers? We want an early, accurate diagnosis so that we can start the treatment as quickly as possible. We want to monitor disease progression and the prognosis, and we want to be able to select appropriate patients to appropriate therapies, which is called personalized medicine. And at the moment, the structural functional imaging, although we might hear differently by Johannes later on, but at the moment there is no PET ligand for alpha-synuclein, so we can't yet see how that is kind of moving along from the body or the brain. And the earlier, more reliable method is really detection at the moment of alpha-synuclein in the physiological fluids, such as CSF with our assay, and then George will tell us about his blood test later on. And the detection of this alpha-synuclein protein that we are doing is really much more sensitive and specific than what has been done before with the conventional ELISAs. I'm going to try to
explain what CSF is.
CSF. CSF is a clear fluid which runs through in your spinal column and around your brain. So that is kind of the closest contact that you have into a brain, and that's why we think that the alpha-synuclein, the proteins that are present in the brain, will also be present in this medium CSF. So I'm going to try to explain this assay, which has a really elegantly simple principle in that sense that we take a patient's CSF sample, which has been taken from you through lumbar puncture. We mix it with human-made alpha-synuclein protein, so this is called recombinant alpha-synuclein, which has been produced in laboratory. And then we also put in there a thioflavine T, which is it's a fluorescent stain. It stains anything, it detects this fibrillization that happens. We start to shake this, we put it in this 96-well plate, all these three in each of these little dots, and then we start to shake. And we shake all the way to the 120 hours. And in the patients, we see that the aggregation starts to occur. And in healthy controls, we see they remain completely flat. And this really is more sensitive and specific, giving us this yes or no answer than conventional ELISA techniques that have measured the concentration of alpha-synuclein, a total concentration, because this is not measuring the concentration, it's measuring the aggregation propensity. Exactly the same thing that happens in Parkinson's brains. So in one of our studies, we wanted to then move on to, first of all, to develop this sensitive and specific diagnostic test for Parkinson's. we wanted to see if this test could stratify for Parkinson's from the MSA. Remember, they both have alpha-synuclein in their brains, but we wanted to see whether the aggregation propensity is somehow different between these two diseases. Then we wanted to measure if we get this kind of yes or no answer, but we wanted to see different kinetic variables. I'll explain to you in a minute what that means, whether they would correlate with the disease severity and the progression of Parkinson's disease. And finally, we wanted to carry out this assay also in patients that have REM sleep behavior disorder. So these patients that are in risk of getting Parkinson's disease. And this is a bit busy slide, a bit technical, but I only want you to pay attention in this first row. So in PD patients, we could see that this test detected the alpha-synuclein aggregation in 89% of patients. In RBD, 65% of patients. Few of our controls were positive as well, but this might just show that, because in some controls you do see alpha-synuclein as well, so that we considered that to be acceptable. And then in 75% of MSAs we could also see this test to be positive. And what I mean by looking at these quantitative parameters is that we get this yes or no answer, that the aggregation happens or doesn't, but then we can extrapolate different kind of things from these curves. We can look at when does the curve actually go up, what time point is 50% of the aggregation happened, and all sorts of other things. And we try to correlate these with the different clinical symptoms and the severity of them. So first of all, we could see that we were able to stratify Parkinson's from MSA. So if we look at this T50, which means that 50% of the protein has aggregated, and the Vmax is just how fast it aggregates, we could see that we could distinguish PD from MSA. And this is just shown here in a different way, these are the plots. Fair enough, we are not able to do this in an individual level. So if we just look at one single sample, we can't say whether that's MSA or PD. But this is interesting because it's showing that the aggregation propensity is different in MSA versus PD. And this could help us to understand more about the disease process itself. Now, when we looked at these kind of quantitative parameters in relation to severity of Parkinson's symptoms, so your motor scores in the UPDRS and all the other components as well, your cognitive scores of MMSE and MOCA, we could not find any correlation. But we were able to see differences between the different clusters that you probably are aware of. Michelle has talked about these many times. So we have 4 different clusters depending on how the baseline motor and non-motor symptoms develop. And in these clusters, with cluster 2 with mild motor and non-motor disease had the lowest proportion of the test responders, whereas the cluster 1 with very fast motor progression all were positive. And then if we finally look at the RBD patients, we looked at 4 different cohorts. So we looked at patients from the Oxford Discovery cohort from Michelle, and we looked at 3 other cohorts as well. And altogether we could see that 2/3 were positive. But this really depended on which cohort you belong to. And that probably depends on how close these patients were to the penile conversion. So they were about to move to become either patients Parkinson's or multiple system atrophy. But during the follow-up, we could detect 30% of our RBD patients that converted to either PD or MSA or another synucleinopathy, and 2/3 of those converters at baseline were positive. So we have to still ask for ourselves why 1/3 that we knew that converted to PD at baseline were still negative. So that is the research question that we still need to address. So I'm just going to conclude here to say that our assay, it is sensitive and specific. It detects 90% of the patients that have Parkinson's disease. It also detects 75% of the MSA patients. And our test does give this binary result but we can extrapolate some quantitative parameters. And these quantitative parameters do help us to distinguish between PD, Parkinson's, and multiple system atrophy. And this is important because the correct early diagnosis and then the prognosis requires this information. And finally, we could not detect in that sense that the assay, our test could correlate with the severity of the Parkinson's symptoms, but it detects differences in these different types, different clusters. And this tells us that the different clusters may actually have different strains. And this is important because if we think about a therapy for just for alpha-synuclein, it won't work. It will have to be really specified to these different strains. And finally, I'm showing you data that our test can also provide, shows promise as an early predictive test in RPD. And I just want to thank everybody in my group and everybody who's helped us to carry out this test and then providing us the biosamples, including Michelle. If you want to read more about this test, please look at these two websites over here. Or you can always email me, I can send you the link on those. And I just wanted to end by emphasizing how vital the gift of brain donation still remains, because as I've explained to you, we have a lot of work still to do to understand these different strains and how we can provide therapies and biomarkers and therapies to really focus on that kind of specificity. Thank you.